Deep inside the human body, the basic chemical reactions required to sustain life are naturally sluggish. Left alone in the chaotic cellular environment, these processes take far too long to occur spontaneously. Every chemical reaction faces a specific thermodynamic barrier. Reactant molecules must absorb a specific amount of initial energy to cross this barrier and convert into a product, a requirement known as activation energy. This graph charts that exact energy barrier. The red curve illustrates the significant amount of activation energy required to push a substrate toward becoming a final product when no biological catalyst is present. Enzymes solve this problem. As the enzyme binds with a substrate, it releases binding energy that pulls the required activation threshold down to this blue curve. The energy cost drops, and the reaction accelerates. The most efficient of these molecules, like catalase and fumarase, achieve a state known as catalytic perfection. They process substrates so rapidly that their operational speed is limited only by how quickly they can encounter target molecules in the cell fluid. If enzymes could not execute this precise drop in activation energy, the biological processes required for breathing and muscle contraction and cellular division would essentially stop. Despite their speed, many of these enzymes do not function immediately upon synthesis. They are initially built as an apoenzyme, which is an inactive, protein-based foundation that cannot drive a chemical reaction on its own. To function, the apoenzyme requires a non-protein attachment. This can be an inorganic metal ion, called a cofactor, or a complex organic molecule known as a coenzyme, which locks into place to complete the structure. With that piece secured, the once dormant protein becomes a holoenzyme. This fully active complex is now equipped with a functional active site, ready to capture and convert incoming substrates. Millions of unique holoenzymes operate simultaneously across the human body, but they are organized into distinct categories based on the specific type of molecular manipulation they perform. This classification table groups them into 6 major classes. They range from oxidoreductases that catalyze electron transfers to ligases that use cellular energy to bond 2 independent molecules together. Early biochemical theories proposed a static interaction, where a substrate molecule matched the geometry of an enzyme's active site with absolute, unchanging precision. X-ray diffraction studies provided evidence for a different model. Under induced fit, enzymes are flexible. The active site adapts its shape upon contact, tightly wrapping around the incoming molecule. This physical adaptation applies tension to the bonds of the substrate, providing the direct mechanism that forces the chemical transformation into a product. Measuring the speed of these physical interactions falls under the study of enzyme kinetics. It defines the environmental rules that control how fast these machines operate. This chart maps enzyme velocity against temperature. The reaction rate climbs as rising heat increases molecular collisions, but only until it hits a specific optimal peak. Pushing the temperature past that optimum peak, or altering the surrounding pH, causes the enzyme to unfold. The protein loses its 3-dimensional structure, destroying its ability to catalyze reactions. The amount of available raw material also dictates the pace. Supplying more substrate increases the reaction rate up to a physical ceiling. Once every active site is occupied, the enzyme is saturated, hitting its maximum velocity. Mapping these kinetic limits allows for the design of drugs that can precisely manipulate these molecular behaviors. When hyperactive or hijacked enzymes drive disease, pharmaceutical drugs are designed to intervene. One strategy is competitive inhibition. A drug is engineered to structurally mimic the natural substrate, allowing it to occupy the active site and physically block the actual target from entering. Scientists track this inhibitory effect using Lineweaver-Burke plots. In this graph for competitive inhibition, the intercept shifts, proving the drug blocks the active site. In contrast, the non-competitive inhibition graph shows a drop in maximum velocity. Here, a drug binds elsewhere, warping the active site so extra substrate cannot restore full function. Deciphering these graphical shifts allow pharmaceutical scientists to calculate effective dosages and predict when a medication might reach toxic levels. Moving from an isolated test tube to a living patient introduces variables at the systemic level. When high doses of these drugs enter the body, the liver's metabolic enzymes are pushed to their limits. This text defines nonlinear pharmacokinetics. a scenario where doubling a drug dose does not result in a predictable twofold increase in the drug's concentration in the blood. This nonlinearity occurs because the body's metabolic enzymes have reached a saturation point. Once these enzymes are overwhelmed, they cannot process the drug any faster, causing levels in the bloodstream to spike unexpectedly. This table identifies specific medications, like phenytoin and certain antibiotics, that frequently saturate these metabolic pathways. Identifying these saturable processes is a necessary step in clinical pharmacy to manage patient safety. Tracing the science from the reduction of activation energy to the management of patient blood levels reveals how biochemistry is applied to create life-saving interventions. Modern medicine relies on mapping these molecular behaviors. By understanding the rules of catalysis, we can accurately direct their operations within the human body.